What we're going to do today is we're going to wrap up the topic of development, and we're going to start on the next new topic, which is movement, which is a big, rich, and interesting field. Give us a lot to think about. I hope you're doing the required reading in the background. I don't need to refer to it in lectures, but you are required to do it. Just to summarize where we're coming from, we started our discussion of development by looking at the distinction between altricial and precocial skills, noting that humans, in common with lots of other animals, but not by no means all animals, are so terribly helpless at birth and require a long period of parental investment. And we saw that this distinction between altricial skills and precocial skills had nothing to do with the intelligence of the animal, or even whether it was like a human or not. It had, instead, we got a perspective. It was a useful perspective that allowed us to recognize that some animals have a time after birth in which these very early developmental processes occur in interaction with their specific environments, allowing a fine-tuning of the match between the organism and its environment. And the insight that we get from this for humans is, of course, that our extremely long period of helplessness is also the period in which we're becoming socialized, because I will emphasize again and again, for humans, the most important element in our environment is other humans. We are social from the ground up, and this is the period, the long period, in which we become members of a particular culture. You can't separate the culture and the biology here. This is the kind of animal we are. We're the kind of animal that is born into a long period of enculturation. That makes culture as biological as respiration. So that's an important insight. Then we looked at these two landmark figures in the study of childhood development. Piaget, with his emphasis on the strict sequencing of particular cognitive abilities, more or less considered devoid from context, but also a strongly biological perspective. And Vygotsky, who took a very different view of human development, looking instead at how we become fully human in interaction with others through the very rich set of social interactions that we have, such that all psychological abilities, from Vygotsky's point of view, are first born, as it were, in an interpersonal public space, in the external world even, and only later become internalized as we acquire the ability to think, reason, speak, and so on for ourselves. But we do that first and foremost in conjunction with others. A view on learning that's not restricted to the first few years of life, but that carries on for the rest of your life, really. And after discussing those two characters, we briefly tore shreds out of the simplistic distinction between nature and nurture. You're beginning to see a theme here, aren't you? Emphasizing that culture is biological. Well, now, in a similar vein, we're pointing out that you cannot separate nature from nurture, that there is no strict biological separation, and that the idea that we could decompose or dissect human behavior or experience along the lines of biology versus education, culture, social interaction, what have you, is nonsense. And then finally, the last day, we met the Human Speechome Project, which was this audacious, ambitious attempt to capture the entire linguistic environment of a child for 3 years, from pre-linguistic to moderately linguistic. The child in question was Deb Roy's first son, Brandon. And the technological requirements of this project were vast, including turning the whole house into a recording center with a massive big data collection server farm in the basement. But dealing with this amount of data is a non-trivial problem. But we saw some beautiful examples. We listened to extracts of the word water as it became a full word. And we saw some experimental results that showed that the child and the environment are in constant interaction with one another, and that the linguistic environment of the child sensitively responds to the child's abilities. We saw that before a given word emerges as an autonomous production of the child, it occurs in relatively simple form in its environment. Caregivers and peers will use shorter, simpler sentences containing that word until the child masters the word, and then the environment becomes more complex from the point of view of that word. An enormously nuanced study that required shed loads of work that would not have been possible before. We saw the rich audio-visual sampling in this example of the ball. Just remind you. Who knows what you can do with this? One thing you can do, for example, one thing that Deb has been doing for the last couple of years is look at the relationship between where words are spoken. So look at the words and look at where those words occur. And of course you find, and you can document from this data set, you can document that the word water will preferentially occur in the bathroom, that there are certain words associated with the hall. There are a different set of words associated with the bedroom. So you can draw a map of the linguistic environment of the child in a completely different way than we would have ever done before. And this work is extremely important because it addresses this old-fashioned and simplistic notion that of the poverty of the stimulus, argued by Chomsky, which suggested that children must be born with an innate capacity for language. What we see here, and it's a constant theme, is that we become who we are through a delicate dance, a delicate interplay with our environment, which includes the environment of other people. That's as far as we got. And we're going to have just one more little foray into the world of developmental studies today. We're going to look at two classics in the study of early infant behaviour, both of them looking at social aspects, again, of early infant behaviour. And what they do is they illustrate some of the rich questions we have in this domain, some of the insights we can gain from studying very young children, but also some of the difficulties that inhere and that necessarily come with working with children. You know that actors say never work with animals and never work with children. Psychologists don't have that luxury. Developmental psychologists have to work with children. So we see in these two studies, we see some really interesting insights into how we become who we are in conjunction with other people, suggesting, of course, that this sociality is right there from the start in our earliest interactions. But we see also the challenge of interpreting human behavior, and there is also, from a scientific point of view, there are great difficulties in replicating such studies. Let's just remind ourselves that scientists very often, where possible, like to stand back from the things that they're observing. They like to not become involved, not to be a participant. So you set up an experiment and you have hands off and you allow the experiment to happen. And then only then do you interact with the data that you collected. So there's this important sense of distance that is appropriate between a scientist and the thing that you're studying. But you know, if you're dealing with infants, you just can't do that. Infants need constant care and attention. You can't be in the presence of a child and ignore the child. You necessarily sort of become involved. And some of the problems of replication come from this necessary link between the experimenter and the subject. And of course, another thing that happens is all babies are really rather different from each other. They all have little personalities. So those are the problems. But here's some of the studies. The first one we're going to look at from 1985 by Murray and Trevarthen looked at the regulation of interactions between 2-month-olds and their mothers. So these are quite young infants. They're only 2 months old. They definitely have no language. And they're interacting with their mother in two conditions. Now, in order to do this in 85, there was quite an elaborate setup. It would be a bit easier today. But anyhow, what they did was they sat the child in a chair looking straight ahead. Now you can see the child here looking straight ahead at a one-way mirror at a 45-degree angle. And above that is a monitor projecting down. So the child actually sees as if the monitor was directly in front of him or her. The child is looking directly at a screen. But it's a one-way mirror, so the child's face is available to the camera, carefully positioned behind the mirror there as well. And the image of the child is beamed to the mother, who's in a different room, who's in a similar setup. So that the mother is looking straight at the child is looking straight at the mother, but they're in different rooms. That's what the setup, why this setup is done. You'd say, why didn't they use Skype? Well, first of all, it's 1985, and you can't use Skype in 1985. Secondly, Skype wouldn't really work because there's all kinds of technological problems. You're looking at a webcam, not looking in the eyes. It's not really like being in the presence of someone. So this is an attempt to make the child and the mother have that impression of being physically present to each other. And each infant in this study was recorded twice. First of all, interacting with their mother in real time. So the mother's in one room, the infant is in the other room, and the mother can't touch the infant, obviously, but can do enough, speak enough, and the infant's looking at the mother, and the infants were perfectly happy to interact with their mothers in this condition. Then in the second session, which was either later on the same day or on a subsequent day, the infant's brought back in, and a recording of the mother from the first session is played. So that what the infant sees is exactly the same as what they saw in the first session. But the infant's response is completely different the second time around because they're not dumb. They realize that they're talking to a recording. I'm sure you've all done this prank on the answering machine where you record an answering thing that goes, hello, hello, hello. You know, it's really annoying. Infants also are not dumb. It doesn't take them long to figure out that they're interacting with a recording, and they show signs of distress. So what we get out of this is that they are exquisitely sensitive to the live presence of the mother, which is a bit of a, though of course they are, moment, right? But it's nice to document something. But bear in mind where we're coming from and the questions that psychologists have been dealing with. Remember the behaviorist approach, which looked at the responses of an organism to a stimulus. So the response was passively driven by a stimulus. The stimulus happened and then the subject responded. Well, here the so-called stimulus has been engineered to be exactly the same on the 2 days. The kid is looking at exactly the same display, but in one case it's happy and in the other case it's not, because it recognizes the contingent interaction that's going on in the live case. It can tell a live interaction with its mother from a failed, phony interaction with a recording. So far, so good, and it's a very important and interesting study. Some studies claim to have found similar effects, so there's been attempts to vary this and replicate it and expand on it. And they've all run into this problem of interacting with babies. Interpreting babies' behavior is very difficult. You need some measure of distress, for example, which is not as easy to measure as temperature. Some infants, of course, will be pathological. They just sit there quite happy to look at a recording of their mother, go, that's my mom on TV. Right? That's the way infants are. So here we can see clearly the challenges of coming up with measures of behavior that are not entirely infused with the subjective opinions of the researcher. So that's Murray and Trevarthen. The next study we're going to look at is older. It comes from 1977. It's by Meltzoff and Moore, called The Imitation of Facial and Manual Gestures by Human Neonates. So these are even younger infants. Neonates means newly born. In fact, they were 2 to 3 week old infants. And what the experimenter did was they videoed themselves interacting, making faces at the baby, basically, and making hand gang signs at the baby. Yo! And looking to see what the baby did. Was the baby already cool? What they found was that the baby seemed to imitate some of the faces that they would make at the baby and some of the hand gestures they would make. And they narrowed it down to 3 facial expressions and 1 hand expression. I think it was the open hand, I forget. You can see there the experimenter on the top and the baby on the bottom. Experimenter sticks his tongue out. Baby sticks his tongue out. Experimenter opens his gob. Baby opens his gob. Experimenter purses his lips. The baby does something similar. The hand is not shown. Now that is fascinating. These kids are 2 to 3 weeks old and they have no experience of ever seeing themselves. How the hell do they know they even have a face? They don't know the word for face. They've never seen themselves in the mirror. They have no context for this, and yet they seem to be responding as if they go, yeah, I'm just like you and I can do that too. To be able to recognize that what you're doing is the same as what someone else is doing, given the difference in perspective, is quite astonishing. But there are problems. The initial study was very carefully done. They videotaped and then went through the videotape frame by frame, annotating exactly the apparent match between the infant's behavior and the adult model behavior. But there are problems with interpreting those. There have been many studies that try to replicate this, and they have not all agreed with the findings of this study about these 3 facial and 1 hand gesture. When something is this important and is also replicated and attempted and we get variation in the results, you have what are called meta-studies in science. These are studies that study the studies that studied the thing. You pool all these studies together, and in 2016 now it looks like the tongue protrusion is probably safe. That is, it is pretty much true that these very, very young infants will stick out their tongue in response to a tongue that stuck out at them, but the other ones don't really hold up. Interestingly, very young chimps have been shown to do the same thing with an adult experimenter. I'm sorry, an adult human experimenter, right? They rapidly lose that. As the chimps get even a little bit older, they quickly lose this. But the very young chimps, somehow, there's enough similarity between the human face and whatever the sense of self is that a young chimpanzee has to recognize the tongue sticking out. I mean, it's not that surprising. You go to the zoo, and if you go and look at the chimps and the gorillas and the orangutans, you'll find a bunch of children there who are making faces at them, right? And hoping that the monkeys, apes, make faces back at them. So, the question of how do we assess whether an infant is imitating the experimenter or not is a really interesting one. It raises what's called the correspondence problem. If you want to describe something as an imitation of something else, you have to say, well, how similar is it? And if you're talking about a baby and an adult, they have different shaped faces. They are quite different in form. The problem becomes more apparent if you consider teaching animals to imitate humans. So if you clap your hands and you get a dolphin to flap its flippers, to what extent is this an imitation or is it something else? Clearly, as the distance in the form of the two organisms becomes magnified, it becomes harder to map from one to the other and say what corresponds to what. So the correspondence problem is always going to be a problem with these imitation studies. But those are two very interesting findings that have generated an awful lot of work, and they illustrate this kind of question that researchers into early human development are passionately interested in, which is how are these infants coming to know themselves in a world of humans? How are they responding to other people? That's it for development right now. We're going to wind up the topic there and move on to our next topic, which is going to be movement. And notice I said behaviour. I didn't say behaviour, I said movement. Even though the subheading there, twitching, running, speaking, blinking, might seem to you to be a description of different behaviours. Now, we mentioned an odd characteristic of behaviour in our first or second lecture. You may remember we discussed Christopher Robin, who had killed his grandmother with an axe and was out busy trying to bury her corpse. And we wanted to describe his behaviour. And we realised that, well, there's no single way to describe it. First of all, we have to choose a time window. So it could be described as behaviour over 1 second or over 1 minute or 1 hour. And even there are multiple ways to frame our observations. We could say he's digging, and we wouldn't be wrong, but we would have missed the obvious thing that he's burying his grandmother, which was also relevant. But we could also miss the point completely and say he's digesting, or we could say he's blinking, or we could say he's worrying. In other words, there's no one way to describe any sequence of movements. Interpreting movement as behavior is something that we do and that makes us part of the business. It's not quite as objective a procedure as you might like to think from science. So identifying behaviors implicates us. So now I'm going to start our consideration of movement with a really tough question. What do brains do? Now, I'm obviously not expecting a comprehensive and full answer to this from you guys. Not for the midterm, okay? Maybe on the final paper. It might seem like a hopelessly misguided question. We attribute so much to brains. We attribute so much to brains that I want to back off a little bit. And I want to ask you not to attribute quite so much to brains until we have a clearer idea of what brains do. Now, when it comes to the heart, we have a really good idea of what the heart is doing in the body. It's pumping blood around the body. It's connected to a series of tubes filled with blood. We know why we have a circulatory system, and we know what the role of the heart is. We have a job description for the heart. We don't have a job description for the brain. If you think of the brain as a controller, the thing that controls your body, this lecture will hopefully disabuse you of that notion. that the brain is not controlling the body, which means we have a big set of interesting questions about what the brain is doing. But let me qualify this. I can provide an answer to this question, what the brain is doing, but it's not going to be a satisfactory one. Supposing your friend Billy Joe is on the phone out there in the hall and he's calling into a live auction in Sotheby's and he's bidding on the Mona Lisa. Right? It's really dramatic. There's lots of money in play. It's a really important thing. But he's bidding remotely on the phone. Now I could look at Billy Joe and I could say what he's doing. And if I were to say he's bidding at an auction in Sotheby's, I wouldn't be wrong. But supposing I said he's on the phone, no, I also wouldn't be wrong, would I? It just wouldn't be a full story. It wouldn't be perhaps a very satisfying story, but we might need more evidence and a richer worldview in order to discuss his being on, taking part in a remote auction, whereas any idiot standing around could say he's on the phone, could see he's on the phone. Well, in that sense of the, yeah, he's on the phone business, what does the brain do? And here we do know some things, because if we follow the brain, we look at the brain, it's connected primarily to the central nervous system, it's connected to the peripheral nervous system, there are nerves whose spikes, whose activity, it goes towards the brain, and there are nerves where activity comes from the brain. And we can look at where it stops. And there's two things that we find. First of all, the brain has some terminal connections in glands, which regulate hormones, which are intricately concerned with the soup of chemicals in the body. And these soup of chemicals regulate, among other things, the brain itself. So that's kind of interesting, but it's also kind of hidden and not something you were thinking of. The role of the brain in modulating, in affecting the chemical economy of your body is poorly understood, but it's one of the things that the brain does. The only other thing that the brain does is to move muscles. So we follow those nerves coming from the brain and they stop in muscles. That's where they stop. And what happens then is that when enough impulses travel down a nerve, a muscle contracts. So in the same sense that we can say of Billy Joe out there on the phone, he's on the phone. Yeah, we're missing the point that he's attending an auction, but he is definitely on the phone. In that sense, brains regulate chemicals and they move muscles. And that's what brains do. When we lay it out like that, we're not going to have much more to say about the chemical economy. I don't know a lot about it, and nobody knows an awful lot about it. We're going to be concerned with the second one, and it becomes clear that movement and brains are going to they're going to have an interesting relationship. In fact, when we look at the evolutionary story, if we look at animals with much simpler nervous systems, and we look at any evidence we have of early stages of early nervous systems, simpler animals altogether, animals even without brains, it is entirely plausible that nervous systems evolved for the principal purpose of allowing us to get around in the world, to locomote, to go from place to place. And moving around is something that very many animals find to be handy. Even some plants find it to be handy. And there are some good reasons why this business of moving around in the world plays such a fundamental role in most animals and a few plants. Food is not evenly distributed in most environments. You can't just sit there like Homer on a donut machine with your mouth open and expect food to land in it. That doesn't work. Even for humans, you got to go down to the cafe and get a sandwich. Resting is very important for very many animals, and even those who don't rest, they don't always eat, and most animals are always in danger of being eaten themselves. So if you're going to rest, if you're going to shut down a little bit, you're going to need somewhere safe to do that. So you need the ability to move somewhere safe to avoid predators. And then the third one is, of course, mating. There's so much activity goes into mating, even among humans, and those are the three main reasons that we move. Food, rest, and mating. So, the close relationship between the ability to get around in the world and the nervous system is nicely illustrated by a little animal, a little gelatinous animal found in the sea called a sea squirt. And we let Daniel Dennett tell the story. The juvenile sea squirt wanders through the sea searching for a suitable rock or hunk of coral to cling to and make its home for life. For this task, it has a rudimentary nervous system. And when it finds its spot, it takes root. It doesn't need its brain anymore, so it eats it. Rather like getting tenure, he says. That's when professors get their final jobs. They don't have to think anymore. They just can pontificate like me. Dennett is hamming it up a little bit here. He likes to do that. Slightly inaccurate. He doesn't just sit down and decide to eat its brain. There's a metamorphosis that happens. This is an animal which has 2 stages in its life cycle, much like a caterpillar in a butterfly stage. And in the process of the metamorphosis, it digests not a brain, but a little node, a little clump of nerve cells known as a ganglion. Now, there's no clear difference between a brain and a ganglion, except that a brain is massively more complicated. Lots of animals, very simple animals, will have bunches of neurons connected to each other, which are called ganglia, and some animals will have very many ganglia within them. With an animal this simple, calling it a brain is an exaggeration. So, okay, we got a little bit of over-dramatization, but clearly this animal, optimized by the processes of evolution, needs a nervous system for getting around and doesn't need it when it's no longer getting around. Nervous systems are high maintenance from the body's point of view. They consume resources. An awful lot of the food you eat goes to powering your brain, believe it or not. So that if it can do without a nervous system, as it appears it can for the second half of its life, it's in its interest to do so. So we're going to study, first of all, how we get around in the world. And we're going to look at it from quite a generic point of view. Locomotion is the business of getting around in the world. And there's a couple of animals. There's a jellyfish, which will undulate slowly in the sea. And there's a bunch of ants. And there's a human with a bug of some sort on his face. And there's lots of different ways of getting around in the world. These are only some of them. You get around with or without legs. Some animals fly, some jump, some undulate. Even those animals with legs, some have 2 legs and some have hundreds of legs. So there's many, many, many different ways of getting around in the world. One thing that's noticed to be absent from here is the wheel. Humans came up with the wheel. Why wasn't evolution clever enough to come up with the wheel? Yeah, any theories? I've never thought of this before. Terry Gilliam, who used to do the animation for Monty Python, couldn't be arsed trying to draw people walking. It's really hard to draw people walking. But stick a wheel on them and it's really easy to animate. Just it does itself. So he used to animate, a lot of the Monty Python animations have people on wheels precisely because they're easy to animate. Nature did actually come up with the wheel. There are certain single-celled animals, bacteria, which have a little device inside them which is a wheel. Now a wheel is not the same thing as a disc or a roller. A wheel is like a disc, but it has a hole in the middle and there's a fixed axle about which the disc rotates. For these small bacteria, this is located at the back of the body of the bacterium, and attached to this wheel are a load of thread-like extensions, flagellae, which when the wheel spins, they rotate and propel the animal forward. It's a means of locomotion, a wheel-based means of locomotion. And it works in an animal this small, but if you try to scale it up, you run into the obvious problem. If the wheel can freely rotate about an axis, and that's what makes a wheel, then you can't have any cables going from the axle or from the surround into the wheel. You can't have blood vessels, you can't have nerves, you can't have any kind of fixed connection because the wheel doesn't accept that. It rotates. It's freely moving. So at a very small scale, the parts of the wheel can be supplied with their metabolic needs by processes of chemical diffusion, which are very slow. It only works at a very small scale. If you scale this up, then there's no way to supply the parts with enough with the nutrient requirements and take away their waste and so on. All living tissue has nutrient requirements. And you can't do it. It doesn't scale up. So nature did come up with a wheel and it didn't scale up. Enough. We move on. Let's move to California at the end of the middle to late 19th century. And this character, Edward Muybridge, he was a pioneer in the early art of photography. Photography was fairly new at this stage, and taking a photograph was a hell of a lot of work. You needed a big camera, and you needed a plate, and you had to stick the plate into the camera, and then you tell someone to sit very, very still, and then you open up the shutter, and they sit still, and then you close it again, and then you go off, and you do a lot of stuff with chemicals, and then you end up with a picture. It's so much easier now, my God, but it used to be really hard. And he was one of the pioneers of this. He developed some wonderful photographic techniques. He did some of the first landscape photography out there. He was the first guy to photograph Yosemite and the fossilized trees and open up the whole wilderness of California. Death Valley, he was the first guy to photograph all these. Edward Muybridge was a colourful character. He found his wife in bed with another man and he shot him dead. And he was hauled up in front of a judge, this being the Wild West. He was hauled up in front of a judge, and the judge said, "Why'd you do it?" And he said, "Well, he was in bed with my wife." And the judge said, "Ah, fair enough, off you go." So he wasn't subject to any penalty. That's what justice was like at the time. Now, I'm not condoning that kind of behaviour, but Edward Muybridge did give us something very, very beautiful. He was passionately interested in studying motion, animal locomotion. And he set up a studio, multiple studios, but his Edward Muybridge's studio was unlike anything else in the world at the time. He would have multiple cameras arranged in series, all hooked up with little tripwires, and then he would arrange for something like a bloody hippopotamus to walk from one end of the studio to the other, setting off the cameras in motion so that you end up with this kind of beautiful diagram. You've all seen these pictures. Muybridge's pictures are all over the place. Turned into GIFs these days, but this is what they were like. And for this image of a horse, for example, he needed this is not one camera, this is 16 cameras in sequence. All, getting the timing and the position right on this is a huge thing. This particular one settled a sort of a barroom bet about the question of whether a horse has all 4 feet off the ground when galloping. You may think you're sure that it does, but you're used to looking at photographs. You're used to looking at video and pause and freezing time like that. Back when photographs were not widely disseminated, it was possible to have a big argument about precisely this question and have no means of settling it. Well, you can see Muybridge settled the case. A horse does have all 4 feet off the ground when galloping. You can stop staying awake at nights now worrying about that. He did beautiful studies, including lots and lots of animals, ostriches, pigs, cows. He did lots of human studies, watching the different way that people carry things, interact, wrestling, fighting, boxing. He did studies of disabled people. He was a consummate, obsessive cataloguer of the details of biological movement. And we share that with them. I think most people will recognize that these are pictures of great value and they inform us. They show us something about how movement works. And we recognize in biological movement, and particularly in human movement, we come to it with a great deal of understanding. Like that baby that sticks its tongue out because it understands something about a face. We understand something about movement, human movement in particular. If you look at this is just a bunch of dots. But when these dots move, you'll be able to, see something. You all saw something beyond a bunch of dots moving, right? You saw people. You saw people kicking and punching. We see that that's an awful lot of understanding that you brought to what was, I emphasize, just a bunch of dots. The dots were, of course, recorded using something called a point light display from someone actually doing those movements, but it just shows how much sense you can make of biological movements, and this is what Muybridge was doing anyway. So, let's come back to the role of the brain in all this beautiful goal-directed movement, and locomotion in particular. I said I wanted to disabuse you of the notion of the brain as a controller. If you think of the brain as a controller, then you're liable to think that the brain is where a decision is made as to where, something like this, that the brain decides where the foot goes next, and then the brain figures out which muscle combination will get it there, and then the brain commands the muscles to move there, and we'd have that cycle. That would be how the brain might control the body. And here is the basic message, and this is the most important thing in this course. I've said it before and I'll say it again. Brains are not people. Brains don't decide. Brains don't figure stuff out. Brains don't see. Brains don't feel. Brains are not people. Brains are lumps of meat. It seems unnecessary to say that, but we are doing science in a context. Science grew up in a society that was largely shaped by Christian theology. And at the heart of that is a vision. This is the underlying vision of Western society, modern Western society, is of the person as completely autonomous, as separated and separable from their surround and from all other people. This is a religious view. It's not a view that science can uphold, but it's still a view that informs most of our brain science, most of our neuroscience, most of our cognitive science. And it's only by making it very explicit like this that we can ever hope to get beyond that. So when you're reading the newspapers, and even in everyday life, you'll constantly see people making the most ridiculous claims about brains. Most of us on the inside of this business are appalled at this constant confusion of people with brains. Have a look at this little guy. This is a puppet. When we speak of the brain as a controller, we're effecting a split between the brain and the rest of the body. Now, there is no such split. The brain is fully as much part of your body as your heart, your lungs, your liver is, right? But we're effecting a split as if the brain were separate, which is just a relationship that the puppeteer has to a puppet, as if the body were the puppet and the brain were the puppeteer. That's what it means to be the controller. Now, the puppeteer here is, of course, a person, so when we treat the brain like this, we're sticking a little person inside the skull, which is not clever. Let's have a look at this puppet, though, and see what would happen if that were the relationship. Well, this puppet has 4 strings. They're crudely drawn on, I admit it, but they are 4 strings. And controlling 4 strings at once would be a problem. It's difficult to do that. And you can see the puppeteer here has helped him or herself out by attaching 2 strings to the different ends of one bar, 2 strings to the different end of another bar, and putting the whole thing into a single assembly so that only one hand needs to move. That greatly reduces the number of degrees of freedom here from 4 to something less, because one string is not independent of another thing. And that's the question we need to ask ourselves if we adopt this view of the brain as a controller. What are these strings? What are these degrees of freedom? What are the individual things that could, in principle, be independently controlled? This notion of degrees of freedom is the single most important concept to get into your mind when you look at biological coordinated movement. If we consider the brain to be controlling everything, well, we have to ask, well, what are the things that it's controlling? And let's consider a relatively simple movement. I move my arm to pick up a coffee cup. Now, there's a myth written on here. I'm glad you're here today, because if you just look at the slides, you'll believe the myth, that there are 7 degrees of freedom in the joints of one arm. Why did I say that? When I move my arm, my shoulder can move in various ways. It can rotate, it can move up and down, and it can move side to side. So those are 3 degrees of freedom in the positioning of my shoulder joint. My elbow joint is much simpler. It's a simple hinge joint. That's 1 degree of freedom. If I was a puppeteer attaching strings, I just have 1 string here for the elbow. My wrist is an enormously complicated joint, lots and lots of bones, lots of interfaces, but essentially it's 3 degrees of freedom, forwards, backwards, sideways, and rotation, much like the shoulder. So that's why it says there's 7 degrees of freedom in the joints of one arm, but the brain is not directly connected to the joints. The brain controls, what we just said, brain controls muscles, interfaces with muscles. So each joint movement is controlled by multiple muscles. Very many muscles are involved in moving even just a simple hinge joint of the elbow. And each of those muscles could, in principle, be controlled independently. So the number of degrees of freedom just went up. And each muscle is composed itself of individual packages or packets. And each of those is, in principle, independent of the other and could be controlled, and is, in some circumstances, controlled independently of the other. And each of those packets breaks down into smaller units, which break down into smaller units, until we get down to the level of the individual nerve connecting to the individual cell. Now, each of those could, in principle, be treated differently, independently, and differently from the other. So the number of degrees of freedom went, now, as we consider the physiology, it went from 7 into the billions. Oh, that's going to be a problem. This is a problem that was recognized in the 1920s in Russia. Some of the finest movement science in the world has come from Russia, and Nikolai Bernstein was a Russian physiologist who did very careful, beautiful, controlled studies of coordinated movements, and he discovered that we never see bodies moving as if they were controlled in this highly implausible fashion. The numbers make this unbelievable to start off with, having billions of things to control independently. That's just a ridiculously difficult way of doing things. The numbers are essentially an infinite number of degrees of freedom. But there's another reason to object to this view of the controller, which is that if that were the task that the brain were solving, then it would show, because we would see lots of uncoordinated movement, and in fact we virtually never see uncoordinated movement in animals or in humans. Virtually never. Even the simplest animals, biological movement is smooth, fluid, and coordinated. You may not feel like that when you're out there playing something, like if you're playing golf or something and you've never played before. You might feel uncoordinated, but it's trivial. You still are massively coordinated. If you ever want to see uncoordinated movement, we do see it. We see it in the twitches of a body in the throes of a grand mal epileptic fit. That's unregulated electrical discharge in the brain, producing uncoordinated movement. So a fit, an epileptic fit, is what uncoordinated movement looks like, and that's the only time we ever see that. So we do tend to think intuitively of all behavior arising from controllers, that there must be a controller there. When we study coordinated movement, we find something quite different. So replacing the notion of control with the notion of coordination will turn out to be helpful. A lot of the insights from this has come from robotics, from building little moving pseudo-organisms. And here's a roboticist, Rolf Pfeiffer, who's going to illustrate the movement of three very different kinds of robots. I hope we can hear this.
Now I'm going to show you a few behaviors. And whenever you observe a particular behavior, the question arises as to what the underlying mechanisms are that bring this behavior about. So now here's one that I like very much Crazy Bird. There's another one. So the question is now, what are the mechanisms, what is the control that's underlying the behavior of these creatures? Now, it turns out that, of course, we know because we built the systems. This is the control. So it's basically just the wheels turning at a constant speed. Okay, now if you remember Crazy Bird, it was doing all sorts of things, but the control is just this. And by the way, the control for all of these three creatures is exactly the same, but they do very different things. So the diversity in the behavior of Crazy Bird cannot possibly be due to the diversity of the control, because the control is, you can hardly call it control, it's just these wheels turning at a constant speed.
So if we think of the controller as that which is initiating, driving the behavior, each of those has just a single wheel turning at the heart of it. And the very different behaviors come about because they have different bodies, because they're embodied differently, and those bodies stand in a relation to an environment. They'll behave differently on stairs or in grass than they will on tabletop. So when we study the form of coordinated movement, we're not really going to get very far if we attribute all the complexity of that to a controller. That's the basic lesson I want to emphasize today, is when we study movement, we need to get away from the underlying metaphor of the brain as a puppeteer, as having an intelligence all of its own, of being like a mini person inside the skull who's doing all the deciding and the feeling and the controlling. Of course, brains are there for, brains play a hugely important role in all behavior, in all movement. In fact, we've just emphasized how the nervous system itself evolved in order to allow animals to move in their environments. But that's not the same thing as attributing the role of the God-given controller to that. When we do that, we make the mistake, the homuncular mistake, of attributing to a brain something that properly belongs to a person. And the person is an organism in a particular environment, as we'll see. So with that lesson, we'll stop right there and we'll pick up the story of movement in the next lecture.